There you go. Okay, guys, welcome to the seventh, whoa, I forgot to change to topic 6. The sixth topic of 285, microtubules. I put this famous quote in by Rich the Kid. I don't even understand how the heck my maps block. Slightly paraphrased, but almost the same. So let's get started. We'll be talking about today the effects of these certain drugs on microtubules. So like in the last review session in the week, Tina and I can't remember somebody else went over all the other materials. So today we'll just be focusing on drugs that affect microtubules. So the first one is Taxol, and this one stabilizes microtubules. And so, like, as you probably remember, microtubules have this property called dynamic instability, where if they aren't adding on GTP-bound subunits, then they will start to depolymerize as the rate of hydrolysis catches up. And so, with this in mind, you know, these drugs right here actually help, or can either help or mitigate this process of dynamic stability or instability. So Taxol is the first one of them, and so it will stabilize the microtubules and prevent depolymerization. And this one is used in a variety of chemotherapy treatments. Colchicine is one that does the opposite, as well as vinblastine. They both destabilize microtubules, and so when they're active, administered to the cells, they lead to disassembly and don't allow polymerization to occur. And then colchicine is used for anti-inflammatory needs, and then vinblastine is used for destabilizing microtubules for chemotherapy. So, I mean, these would be a nice little thing to write on your cheat sheet. You know, you don't have to write like a whole thing about how they use stuff in medicine necessarily, but knowing whether they stabilize or destabilize microtubules is definitely key for if you stumble upon a question. So continuing on now, we have so we have MAPs, which are microtubule associated proteins, and they can also, like, I don't know, help or not help. So tau is a very famous one, and I believe this is the one involved with Alzheimer's disease. And so tau is it's not a drug, so it is found in the cells, and when it's working normally, it's in the neurons of the central nervous system. And so the microtubules, as you'll see in the future slides here in this presentation, they're used as tracks to move molecules around the cell. Like everything we talk about getting moved around the cell, like all of these are like vessels and stuff are transported through these networks, these complicated networks of microtubules. They're extremely important to the cell. And so tau binds to the microtubules to help stabilize them. And the problem is when it's dysfunctional, though, it will cause disassociation from the microtubules. And basically when it has this effect, or when it has this mutation, it ends up leading to the depolymerization of these microtubules that are supposed to stay stable for the cell to use. And this can lead to tangles in neural cells and as well as it's thought to be the primary cause for Alzheimer's disease. So there's a lot of research, a lot of literature you can look at and read about how tau um, mechanisms for how it could possibly cause Alzheimer's disease, which is a pretty amazing application of your knowledge in this class. So now we'll move on to motor proteins. And so for all of these that we'll talk about, they're all ATPases. So we've had a lot of GTPases in this class. And if this is not like that, it uses ATP instead to direct the motor functions or like the movement, like you saw in the very first slide with the big quote. All of that movement is fueled by ATP, every single movement. And so diamine and tyrosines are the main types that you'll learn about in this class, which they're using eukaryotes, and they help move all these like vesicles and cargo and stuff around the cell using the microtubule tracts that are super important. So, yep, yeah.
Or I can also.
No, you're good. You're good. I just needed to check here real quick. Yeah, what's your question?
So I was a little bit confused because I watched movie 1727 and it talked about like Hydrogen ADP binding for the motor head with ADP binding and then ADP dissociated with the bond and ATP fusion and then this positive to negative like this catalytic before. So I have kind of like a couple questions about that. But one was, so is the nectar zipping onto the actual like, was it zipping the motor head to the microtubule more tightly? I didn't get the zipping.
I believe the zipping you're referring to is just like the movement of it onto the microtubule, like the head onto it.
Okay.
Does that make sense, or is that not what you're saying?
So yeah, so then I was kind of wondering like, then I talked about when it hydrolyzes ACP to ADP, kind of the next thing for relations. But I was confused because I need to ADP still have a delivery for.
No. So as soon, well, no, as soon as the hydrolysis happens, what happens is that if I'm remembering this correctly. Yeah. So when so when the ATP is bound, it has a good affinity for the microtubule. And then and then the binding of the ATP causes like the nectar to move, but once it moves, then it no longer has, then like the ATP, the third phosphate dissociates, it's hydrolyzed, and then once it makes contact with the microtubule itself, it no longer has affinity for the ADP, and then that'll dissociate away.
Right.
So it's like changing conformations. Okay. It's the same like idea as like when you have like the cargo proteins with like nuclear transport, and like you have like something grabbing onto a molecule, and then when it's attached to like an ATP, it changes conformation, and then that changes it to like have like an affinity for something else. It's the same thing, like dissociated away. It's the same concept, just with the movement. Does that make sense at all?
Yeah. There's part of my question is that
Oh, okay, gotcha. Yeah. Okay, so moving on, we'll have the diamonds here. So these ones they're all multifunctional, like minus ended directed motors. kinesins have a bit of a variety, they tend to be plus-ended, but, diamonds are all, minus-ended directly motors. Kinesins, on the other hand, they have like a whole variety that you have to know about it. They tend to go to the plus end. But there's one exception with the 14 family, which is not, it's minus undirected. And then, kinesin 13, don't have like a directional walking movement, and we'll get to that later. Oh. Yeah, so each one of the motor proteins has like a tail up here, and then the heads that are attached to the microtubule itself. And this is where all the walking is going. That was like where all the ATP binding and hydrolyzing occurs that we were just talking about. That actually drives the movement of the heads. So kinesin 1 is the first one we'll talk about. It's called that, the first one, because it was the first one discovered, and it's used for transporting proteins and cargo around the cell. It is a dimer, so it's actually a pair of kinesin monomers, and it's also a dimer when it's inactive as well, so it's like the head and the tail. And so as you can see, like the tail portion right here is attached to the cargo, and then the head will be attached to the microtubule itself. And the head will, be walking towards the plus end typically, or not typically, kinesin 1, so it is going to be walking towards the plus end, but also on the tail end, keep in mind, these are not walking. The tail end here is just holding on to the cargo, the cargo, sorry. So, yeah, there's a pair of heads in the microtubule, and then a pair of the cargo, and they're moved in the plus end of the microtubule. Kinesin 3 family, also dimers, but they're monomer in the inactive state, and they are also plus injected. They're just used for something slightly different. They're used for cargo transport in neurons, and specifically the movement of synaptic vesicles. So between neurons. And then kinesin 13, also a dimer, but they slide rather than walking along the microtubule. They diffuse laterally towards the plus end. But the big difference with kinesin 13 is they have the ability to hydrolyze and depolymerize the microtubule. And this is really key. It will be key for next week when you guys learn about how the cell progresses from anaphase to, from metaphase to anaphase, and how the chromosomes come apart between them. So yeah, you'll learn about that next week. But basically, for this week, all you need to know is that as they walk along, they will depolymerize, and so it would almost look like it's being pulled along a string. If you look like the minus end on the plus end, like it's walking towards the plus end, it's depolymerizing, so it almost just looks like it's staying still at the end, and it's like it's walking, or it's like it's being pulled, because you're seeing this disappear. Kinesin 5 family is a jumbo shaped tetramer rather than diamond. This allows it to have, so first, it's got 4 subunits. So the other ones usually had just like a pair at the bottom and a pair at the top, and depending if they were, in the inactive state or active state, some of them changed to maybe a monomer. This one stays touching it at both states, but because it has like a tail and a head at either end. This allows, it's almost like having two kinesins attached to each other. So it allows for two microtubules to slide along one another with respect to each other. So it's not like, it doesn't really look like it's moving, but they're both walking towards the plus end of a microtubule, which just causes like, when a, so when a motor person is walking this way to the left, it's causing the microtubule to go this way. It's like if you were playing tug of war and you were like pulling, your hands would look like they're going like this way on the rope, but the rope would actually go this way. So it's like the opposite direction. And so what it can do is it can actually, it helps stabilize these microtubules, but it also helps to regulate the movement of them, as it like pushes them like and pulls them apart. And you'll see in the next week's topic about how it does that exactly, and why it's important to the regulation or the execution of cell division when it comes to pushing and pulling a cell into two different cells. Yeah. Oh, I forgot to change the last slide, but if anyone have any questions? Okay. All right, then I will stop the recording here unless anyone has questions here. I am in your 426 class. That's funny. Okay. All right. Does anyone on Zoom have any questions?
No, I don't have any questions.
Okay, cool. All right. Happy Halloween weekend. And how do I stop sharing this video? Yeah, I can't find the screen. With the grandpa right now. Yeah, wait. Can you come over and help me? I like literally can't find it. Oh my god, it's another screen. Never mind. Sorry, yeah. There we go. Okay, if there's no more questions, then I'm gonna finish this.